Hi, this is Ms. Litton, and this is Honors Bio Review Unit 6 for your final. If you weren't here last time, I just want to remind you that on our Honors Bio homepage, you have all the calendars for the entire year. Obviously, you only need to pay attention to 5, 6, 7, and 8. As part of Unit 6, we had Chapter 24, which was DNA structure and replication. The second half of Chapter 24 was biotech, and that was part of Unit 8, but I'm just going to review it as part of this chapter, just as one whole chapter, okay? So we're going to have a little smidgen of Unit 8 in this review as well. I will make a PDF of this presentation. In fact, I already did, and just like I did yesterday in Google Classroom, where I put it up, the PDF, and the YouTube video, I'll do the same thing today. So if there's something in here and you're trying to type, you don't type fast enough, just know it will be there. All right, so before we even start, I want your brain to start to reach back into its recesses and try to find in your brain the structure of DNA, how we know it's the structure of DNA, right? There were some experiments. The structure of DNA, and then talk about the process of DNA replication, transcription, and translation. Because this whole unit is about those things and how it is controlled. So before I start, take a minute just to confer with whoever you're sitting next to. What do you remember about the experiments of how we know DNA is our genetic information? What is the structure of DNA? What do you remember about DNA replication, transcription, and translation? Go ahead and have that talk.
Easy go. DNA structure, I'll give you that, you can read how it's found, alright? So you have the phosphate group on the side. You have the phosphate group with the nucleotides, which was
Here, I can
Okay, I forgot to hit pause. Sorry about that. We're going to start right now. Okay, guys. Hopefully you remembered a few things. So let's go through it. Here are the big ticket items that we did discuss in Chapter 24. The history of DNA, that we know it's our genetic information, its structure and replication. Gene expression, remember, is you use the DNA to make complementary mRNA, and that's going to happen inside the nucleus. And then translation is using that mRNA to build a protein. Remember, there were 3 kinds of RNAs. Do you remember the 3 kinds of RNAs? mRNA, tRNA, and rRNA. M stood for, and who did it get its message from? DNA. rRNA was part of a ribosome. T stands for transfer. What does it transfer? Amino acids to the workbench, which is a ribosome made out of rRNA and proteins. Mutations, when good DNA goes bad, right? All different ways to mutate and different types. DNA technology, recombinant DNA. What do you hear in recombinant? Recombine. Did you recombine some DNA? Yes, you did. Almost all of you had evidence that you at least gave it antibiotic resistance, and some of you even got it to have a gene where you took a silent gene and got it expressed, the RFP gene, yes? What was your evidence that you were able to do that? The plates, the growth on the plates, right? Okay, and so we'll just review the different steps of that. We didn't do PCR in this lab, but we will talk about that as well. All right, here we go. So do you know what that is right there? Bacteriophage. That is a virus that infects what? Bacteria. When they were first studying these, Hershey and Chase, sorry, it's going to bug me. When we were first talking about this, remember, they did not ever see what it looked like, a bacteriophage like you just saw in that microscopy. They didn't know. They just knew that a virus contained, part of it was a protein and part of it was a nucleic acid. So they knew the structure. They knew proteins were built out of amino acids, and they knew that nucleic acids were built out of nucleotides. So what they did is they used S35 to label an isotope to label the protein, and then P32 to label the nucleic acid. So the virus parts were labeled. They allowed the viruses to infect bacteria, and then they looked to see what's inside that bacteria. Did it have radioactive sulfur in it? If it did, then what was transforming the bacteria was a protein. But if it had radioactive phosphorus in it, which it did, then you knew what was transforming the bacteria was nucleic acid. So that was one of the historical experiments. Also, you want to know about Chargaff. Remember, Chargaff said, however many, when you analyze DNA, however many adenines you have the same amount of thymines, and however many guanines, you have the same amount of cytosines. Okay? Also, you remember Rosalind Franklin? She did the X-rays. She would shine the X-rays through crystallized DNA, and the pattern that it generated, and I don't think I have a picture of it, maybe. The pattern that it generated, they knew that whatever DNA was, it was in a helical repeating pattern, that they could see that of the same diameter across the length. So Watson and Crick were able to use all of that information in order to come up with the double helix structure. It's a twisted ladder. What holds the two sides of the ladder together?
Hydrogen bonds.
Hydrogen bonds. What's along the sides of the ladder? Phosphate sugar backbone. Good. All right. So we already know this, a purine with a pyrimidine. Remember, the purines are the two rings. If your nucleotide or your nitrogenous base has two rings, it's a purine. It's a pyrimidine if there's only one ring. There's two hydrogen bonds between adenine and thymine, and there are actually 3 hydrogen bonds between guanine and cytosine. Right? As far as the replication of DNA, it's called semi-conservative replication. That's because each old strand, if this is your original DNA in a cell getting ready to do, let's say, mitosis, during what stage of the cell cycle would they be doing DNA replication? Synthesis stage, which is between what 2 stages?
G1 and G2.
Good, you're so smart. So it's called semi-conservative replication because each old strand serves as a template for the new strand. So if one of your old strands has an adenine on it, then a complementary nucleotide brought in by DNA polymerase 3 would be thymine, right? Guanine with cytosine. And so when you're all done, you have 2 copies. What would these be called right here? Sister chromatids held together by a centromere, which separate during what stage? Anaphase. Okay? So these are your sister chromatids. So each sister has an old strand and a new strand. That's why it's called semi-conservative replication. If it was conservative, the 2 old strands would reunite after being a template, and the 2 new strands would get together. All right? Can you check with the person next to you and see, is there any part of that that's kind of scary to you that you need to jot down, that you need to review some more? Go ahead.
Okay, so there's adenine, thymine, cytosine, guanine, held together by hydrogen bonds. The backbone is phosphate. Just scientific function.
Okay? A little reminder when you compare DNA with RNA. DNA has the base thymine, where RNA has the base uracil. DNA has the sugar deoxyribose. RNA has the sugar ribose. DNA is double-stranded. RNA is single-stranded. Perfect. Okay, so the next part we're talking about is using that DNA to make RNA. That is called transcription. This is a eukaryotic cell. How do I know it's a eukaryotic cell?
Double-stranded DNA.
Because you can see the nucleus.
I can see that I have a nucleus, right? I have a nucleus. Remember, eukaryotic cells have a nucleus, right? They have membrane-enclosed organelles, whereas prokaryotes do not have a nuclear envelope. So as soon as they do transcription, they can immediately do translation. It is not compartmentalized. So here you're using your DNA to make now a single strand of mRNA. So you don't talk about it being semi-conservative because only one of the strands is used for a short term just to get the bases right in your RNA. And then once you have the bases right, it leaves and the DNA zips back up again. Remember that part? And wherever you want to bring in, if you have a G in the DNA, you would bring in a C. If there's a C, you would bring in a nucleotide that has a G. If there's an A, you bring in one that has a U. But if there's a T, you bring in one that has a A. Okay? And it serves as a template. That's transcription. When this RNA leaves the nucleus and it goes out and gets on a ribosome, that part is called translation. Now we know that it's not as simple. When RNA polymerase, it has to get on the promoter and things can inhibit that. We know once the RNA is made, it has to have a cap of 7-methylguanosine and a tail, 100 to 200 adenines. I don't believe I had you memorize what the cap and tail were. And you have to cut out the introns before it leaves the nucleus. Do you remember all of that part? Okay, so there's processing that has to take place. Then it leaves the nucleus and it goes out and gets on the ribosome. These guys right here are called tRNA. T stands for transfer because it's transferring amino acids. When you hook amino acids together, what do you have? Protein. So that brings us to the central dogma. Okay? DNA, just making more DNA when you're getting ready to do mitosis or meiosis, that's just called replication. When you use that DNA to make an RNA strand and use that RNA to build a protein, then the DNA is dictating what that protein is via the mRNA, right? And using that message to know the sequence of amino acids you want in your protein. That is translation. So this right here would be the genotype. This protein would be the phenotype. You see that from yesterday? So you have your genes here, but how it appears, okay, is the phenotype. All right. So this is right. See if whoever's sitting in the blue seat, see if you can explain what's going on in this picture right here. So your original DNA is up there at the top. That DNA has been transcribed into mRNA, the primary transcript, where if this is a eukaryotic cell, we're still in the nucleus. We're going to put on a cap and a tail to protect either end. And then right here, E's and I's, E's and I's. What do the E's and I's stand for? Introns and exons. And we don't especially like their names, right? Because the exons are actually kept, and that stands for being expressed. And the introns are actually what? Cut out. Yes?
So AJ actually sounded like a good thing to remember. He said, say it for you. He said introns are in trouble.
I like that.
That's a good
I
one,
like that. His whole, why did you tell me that? Introns are in trouble. So they are getting cut out and the exons are going to be expressed. I like it. And that's why you can have one gene and multiple proteins from that one gene, depending on what exons. In this case, we have, if we want to call this exon 1, exon 2, exon 3, they are still here, right? Exon 1, exon 2, exon 3. But if we had cut this one out, this would generate a different protein, right? If we just used exon 1 and 2, or if we just used exon 1 and 3, but it all comes from the same DNA. Okay, next. Talk about Slate, whoever is in the Slate. See, talk about what you see here. See, I've reviewed it a little bit, but now you try. All right.
I don't know if this is important, but would you say that the codons code for the anticodon or do they code for the amino acid?
Okay, so let's talk about that. All right. So we've said DNA is the code book for all of your proteins. So how does it do that? Well, the DNA is not going to leave the nucleus. It's like your hard drive of your computer, right? But you can get some information or copies out of your hard drive. And so in this case, that information is your mRNA. Now your mRNA is complementary to your DNA. So they're not going to line up and match like the same exact letters, right? Because it's making what's complementary to it. Where there's an A, you're going to have a U. Where there's a G, you're going to have a C, right? Then you use that mRNA, and every 3 bases on the mRNA is called a codon. That codon dictates a particular tRNA. an anticodon, right? So if the codon says CCC, tell me what the DNA was that coded for that. GGG. Tell me what the tRNA is going to be. GGG, right? Because the DNA says GGG, the mRNA is going to say CCC, every 3 bases, right, is a codon. The tRNA that lands, if this is GGG, this is going to be what? CCC, right? That's going to be complementary to it. The anticodon is carrying the amino acid. What they have done is they've made a chart. The chart that you see in the top right-hand corner is a codon chart. So they know that if you have this particular codon, this is the amino acid that the tRNA is going to carry. So you use that chart. So you could say, if my codon's this, I know what amino acid is coming. So that's the message that you're going to be looking at. And the reason is, not every codon codes for an amino acid, right? You have stop codons that are in there as well. And those stop codons don't code for anything. It's just it's 3 different ways to end the formation of that protein. You also have an initiation codon every single time. It's AUG. Now that may not end up being in the final protein product, because in the chaperone proteins, when they're folding your protein that you just made, they may cut it off because sections of it can be removed. Okay? So the anticodon's on the tRNA, the codon is on the mRNA. All right, you can see the same thing again. Whoever's turn it is, I think it's back to blue. Why don't you take a shot at this picture? Try to put it ayao in your own words. Go. Less time. I'm going to give you less time.
Which is connected
Okay, can you do a check with your bio buddy and just see, is there anything you're not getting right now? Ask them. Is there anything you're not getting right now?
So things that might be so tax locations.
Okay, now, I can make it sound very simple because this is the 30th year I've explained this, okay? 30 years of explaining this, okay? I understand if it's not hard for you, okay? Ask those questions that you might have. Because you might understand it when you're with me in the room, but then you go home and like go, I don't know what she was doing. Yes?
Should we know about the A site, the P site, and the E site?
I haven't got to that part yet. Okay, but I'll go over that. Yeah. Anything else on this part? Okay. All right. Next. Maybe I'm not getting to this part. Maybe we will need to answer your question right now. Let me look. Yeah, I am going to get to that. Let's talk about that just for a minute. The A site, thank you. The A site is where you have just a tRNA landing with just its one amino acid. The P site is where you see the protein building. That's how I think of it, is the P for protein. And then it's not showing it to you on this particular ribosome, but there's an E site. What does the E site stand for? Exit. It's the tRNA that's empty now. And so remember, as each new tRNA lands, the new guy who lands in that A site, as the ribosome shifts, then the chain of amino acid that is connected to the tRNA in the P site, he gets dumped on the new site. And then the guy that was in the P, the protein site, gets moved to the E site, right? And then the guy who was in the E site, he's hasta la vista. Yes?
So the A site is wherever the amino acid is, not just the first one?
The first one does go in the P site, even though it has a single amino acid. But from there on out, every new one that lands will always be on that A site. And that's how I remember it, is A for amino acid. There's just a single amino acid, but the P site has the long protein on it. Okay, more on that. Good question. All right, so let's talk about when things go bad, and those would be mutations. Errors during replication. When you do DNA replication during the S stage, remember how we have those stops where we check to see in the cell cycle if everything's going all right? Usually those are repaired right away, but sometimes an error can go through. If you have a mutagen of some sort, which of all of these, I would say UV rays from the sun, radiation from the sun would be most likely. That's why you wear sunscreen. Just don't smoke, and then that won't be an issue for you. So that's when some sort of chemical can destroy your DNA. And then transposomes, you don't have a lot of control over that as far as we know. Do you remember what transposomes are? Oh, I said it. Jumping genes, yeah. That's weird to me. They just excise themselves out and jump in somewhere else. And then Indian corn was our example of that. And there it is, transposomes. Frame shift mutations. If you get this mutation, it is really bad. Because we know we read in frames. Every 3 bases is a codon. So if somehow that gets off by 1, then every codon we read is wrong. And the example we did is we did 3-letter words. The big fat cat ate the big rat. And we understand that. But if this E in this 3-letter word is eliminated, all of a sudden the very first word is not going to be T-H-E. What is it going to be? T-H-B. If attic atatet have a at, right? And that throws all of them off. It makes no sense because every codon from then on out is broken. So that by broken, I mean that it's going to code for the wrong tRNA, which is going to then bring you the wrong amino acid. Okay, so frame shift mutations, very, very bad. All right, cloning. Now, let's talk, and this is jumping up into Unit 8, but at least this part of it we'll already have done. Cloning is when you make lots of identical copies. So you can do that in the lab situation. You could clone a single gene, like if you used a thermocycler, bless you, PCR, or you could, and you did this, you made bacteria that were clones, right, of a bacterium that was resistant to an antibiotic, or it could make the red fluorescent proteins. Those are going to be identical clones, copies of the original. What we worked with was a plasmid, an independent piece of RNA from the bacterial chromosome. You cut and you splice. What did you use to cut your plasmids? Restriction enzymes. Restriction enzymes cut at a specific series of bases called a what? Recognition there's your cloning. Called a recognition sequence. Now, we like it when they come in and cut across because that creates what? Sticky ends, which allows us to mix and match different DNA. And that's exactly what we did, right? We cut the RFP out of the pecan, and we cut open our PR and cut a small section out of that. Since it was cut with the same 2 restriction enzymes, then we could mix and match the DNA. And what would we have to use to seal? Hydrogen bonds would hold the T's with the A's, but what would we have to use to seal the phosphate sugar backbone? Ligase. Remember, that's the ligase I gave to you at the very bottom of your tube, and then you added everything into it, right? But the ligase was sitting in there. Polymerase chain reaction. Polymerase. Poly means many. So this basically forces what happens in a normal cell cycle during the S stage. It forces it multiple times to occur. And it does that through a series of heating and cooling cycles. That's why it's called a thermocycler. And it has to provide in the thermocycler, you have to have enzymes to make it happen. And we use, I don't remember if I talked about it in honors bio, I don't want to go all AP on you, but Thermus aquaticus, did I talk about that? Okay, never mind. So anyway, you have to use a DNA polymerase that can work at high temperatures, and you have to provide it nucleotides. And then as long as you go through the heating cycle, and the heating cycle denatures it, like, remember what we used to unwind and unzip? Do you remember what its name was? Helicase, right? So you're using temperature to do what helicase would do. Because and then once you've undone it like this, if the heating part would wreck that particular enzyme. And so that's why you force it every time by heat. And then the DNA polymerase will use the nucleotides and make complementary. So you go from 1 strand of DNA to 2, and then 2 to 4, just through the heating and cooling. All right, gel electrophoresis, another tool of which you've used multiple times. Once to separate inks and once to separate your DNA. You know that your DNA, if you put it in little well holes, remember how good you were at that, putting it in right in there, and then you turn on what to make the DNA move? Yeah, the electric current, and then the DNA would migrate through the gel, and the smallest pieces would move the fastest, right? And the smallest plasmids would move the fastest. And then you could compare it to a marker to see, did I cut it in the right place or did I ligate it? There's another gel electrophoresis. Before I go any farther, can you ask your bio buddy, how are you with that? How do you feel? Check with them. Anything you have questions about? Yes. I need to hear the question.
From before when we talked about the causes of mutation,
Yes.
Would those be found in the controls section? Like, you know how there's stages of control throughout the cycle.
Oh, you mean would it be found out and fixed, or what do you mean? I think we usually find any mistakes in DNA replication are usually found and corrected, but it's when they aren't found and corrected, as evidenced by cancer and the proliferation of that, right? Those are one mistakes. And that's because, remember, we'll talk about those two genes that are remember when we talked about oncogenes, right? They were proto-oncogenes before that, but they're the ones that say go cell cycle, go cell cycle, go. Remember that? If those mutate, it just has a whole bunch of problems as a result of that. And tumor suppressor genes, remember, if a cell is out of control, they're the assassin who goes and kills it. So if that is also broken, that one gene doesn't get repaired, then you have a whole onslaught of different cancers. All right, so any other questions you want to ask me about that? Okay, transgenic cat, trans change in the genes, transgenic cat. In this case, you've altered the DNA in the zygote somehow in order to make it so that you're not allergic to it anymore. And so when we look at what is a transgenic organism, it's organisms that have had a foreign gene inserted into their own. Okay, and that's the end of that. Chapter 25 is all the controls of gene expression. And when we look at those controls, here is a really go-to big ticket page. Proto-oncogenes and tumor suppressor genes. We have talked a little bit about that just a moment ago. We said, what is the job of these 8 things? And then what are the 2 main causes of cancers? What are ways to diagnose it? And I'll go through it with the review, but let's see what you remember, okay? A promoter. What is a promoter is found on DNA. It is a binding site. Do you remember what it's a binding site for? RNA polymerase binds to the promoter. This initiates the transcription. If RNA polymerase cannot bind to the promoter, then no transcription takes place. No mRNA is made. A regulator gene. A regulator gene codes for something. Do you remember what a regulator gene codes for? A repressor. Okay, so then that brings us the idea of a repressor, and that's number 5 on our list. What can a repressor do? It can bind to the operator. Now, why do we care? Operator, but the operator sits right next to the promoter. So when you have a repressor sitting on the promoter, it usually kind of spills over and blocks the, I don't know what I said exactly. If a repressor is on the operator, it spills out and then it can block the promoter as well. And I'll show you a picture of that. Structural genes or whatever you're coding for. You know what RNA polymerase is. A transcription factor. Do you remember what that does? A transcription factor will help RNA polymerase to bind on. That's like your Boy Scout. Whereas a repressor gets in the way, a transcription factor will help RNA polymerase. And transcription activators help the transcription factors. So let's take a look at that on a diagram. These are all different levels of control. What we just talked about right here in this is transcriptional control. And this is the superstar of control. This is where most control takes place, going from DNA to RNA. Previously above that, where it says chromatin structure, this is the DNA unwound? Is it even available for transcription? If you remember the talk about epigenetic inheritance, epi means above the genes. So let's say you could have a gene like I am physically a girl, right? But let's say I have a Y chromosome in all of my cells. And that Y chromosome was never allowed to be expressed because the DNA was tightly wound. So even though I have that Y chromosome, since it could never ever be transcribed and it was tightly condensed, I never expressed it. That is called epigenetic inheritance. That's not true for me, but it could be true for people. That would be an example of epigenetic inheritance. Transcriptional control. Is RNA polymerase able to get on the promoter? Post-transcriptional control. Now that I have an mRNA, remember we already talked about this today, you have to put on a what? Cap and a tail and cut out the introns before it leaves the nucleus. That's post-transcriptional control. And can it leave that nucleus? Translational control. Is the mRNA able to get on the workbench? What is the workbench? Ribosome. Okay. Is there some other kind of antisense RNA binding to the mRNA inhibiting that? What, is translation able to go forward? Post-translational control is, now I have my protein. It's the beginnings of that protein. It's just a strand of amino acids. It has to get folded into the correct shape before it can do its job. Post-translational control is getting that chain of amino acids in the correct shape so it can do its job. Maybe it's an enzyme and it needs to have a particular shape to be an active site in order to catalyze a reaction. That would be an example of post-translational control. This is what we've talked about. Here's a repressor. It was coded for by a regulator gene. Active repressors go and get on the operator and turn that gene off. That's not a bad thing if you don't need the products of the structural gene. Right now, in your bedrooms, in your homes, all your lights should be what? Off. They should be off because you don't need them. Your light needs to be on when you go home and you're reviewing or reading a book or whatever you're going to do in your room. Then you turn your light on when it's needed. When do we need, if we need this product, let's say it's the enzymes to digest lactose. If I'm not eating lactose, I don't need those genes on. But if lactose is there, lactose can bind and be an inducer and remove this repressor so that RNA polymerase can bind to the promoter and turn on the genes in order to make the enzymes to digest that lactose. And when all the lactose is gone, turn the gene back off again, right? Because there's nothing to pull the repressor away. Let me show you another picture of that. And whoever's turn it is, talk for just a moment. And this is your example of what I just talked about. Go ahead, see if you can put it in your own words.
But lactose can be produced to take off that.
Okay, now, this one just shows a little bit more detail. The regulator gene is on the regulator gene's coding for repressor, which is on the operator. And so RNA polymerase cannot get on the neighboring promoter right here. Here, lactose is present, so now I need it digested. This is a great way. When I need it, then it turns that gene on. It's kind of like if you walked into your room and automatically the lights turned on, right? So your presence does that. Lactose binds to that repressor, makes it inactive. Now RNA polymerase can get on the promoter. The genes are transcribed. We have the mRNA can be translated into enzymes that will in fact digest this lactose. And then when the lactose is gone, then the repressor can get right back on the operator and switch all of this off. So it's just acting as a switch. We're going to go right through that. Transcription factors help RNA polymerase to bind on snugly to the promoter. And activators help the transcription factors. When you're wondering, hey, is this DNA active or not? Heterochromatin is dark staining. The reason why it's dark staining is it's all coiled up really tight, so it's easy to stain. DNA that is not coiled up, that means it could actually be being transcribed right in that moment. Now, a really good example of an inactive gene that you can see is called a something body. Do you remember what that is? Bar body. Who has bar bodies? Yeah, females have bar bodies because we have 2 X chromosomes and a different X will condense in each one of our cells because we don't need 2 at a time. In fact, that can be a problem. All right, and here's an example of bar bodies. Transcriptional control. This is just kind of, we already went over that, so I hope you're okay with that. Here is showing you post-transcriptional modifications, cap tail, cutting out the introns. Translational control would be, can I build that protein on my ribosome? And post-translation is any kind of modifications that you do to that protein to make it functional, folding by the chaperone proteins. I'm skipping through that. Cancer, when good cells go bad. You can tell looking at a cell under the nucleus if it has an irregular nucleus, extra chromatin within that nucleus, multiple nuclei, those are all signs of cancer. They defy normal cell regulations, and if they, do you remember what the word is here when they can invade other places? It starts with an M. Metastasize. Yeah, and it can move to other places in your body. Another horrible thing about cancer cells is normally they just run out of food because you had no planning to supply that many cells with nutrients to provide it oxygen and take care of waste. But what cancer cells can do is a process called angiogenesis, which is the beginning of blood vessels. They can cause blood vessels to grow towards the tumor and supply it with the nutrients and remove its waste for it. So any kind of drug that would inhibit angiogenesis around tumor cells would be good. Proto-oncogenes are part of the cell cycle that say go cell cycle, go cell cycle, go. And they are in a point of control, like a general of an army. If the general goes bad and that general controls that entire army, right, then he can make them all go bad and all become White Walkers, right? Okay. RAS is like stepping on, obviously not enough Game of Thrones people here. RAS is like stepping on the gas, and so this gene is one of the ones that tends to go bad typically, and even though it has not received a signal, if it becomes an oncogene, it's not received a signal to induce cell replication, it still triggers it. So that's how I remember that RAS is like stepping on the gas. A tumor suppressor gene. What these guys are is the fail-safe. If you have a bad RAS, if you have cells growing uncontrollably, he's supposed to take them out. He's supposed to take care of that DNA, and P53 is a really good example of that, and it's supposed to wipe out those cells. Those would be like the assassins. But if your assassins don't have live bullets, if they cannot take them out, then that's when you have a problem, because then it's like if somebody, remember I gave you the analogy, RAS is like stepping on the gas. If you are learning how to drive and your bio buddy who is riding in the car with you steps on your gas with their foot, and you're like, what are you doing? At least you still have your what? Brakes, right? But if your brakes fail as well, then you're going into the intersection. And that's what we would say is our analogy for cancer. And P53 gene is that gene that is like the assassin. Causes of cancer, you can inherit it. Like the breast cancer gene, people go to look to see. You could also get it from your environment. Overweight, smoking, being out in the sun without sunscreen, chemicals in your environment are known carcinogens, certain chemicals. Okay, so, oh, we did it! Done. I am so proud of you, okay? Done. Okay, so we have reviewed Unit 5, we reviewed, it didn't work. And now we are, and 5 and 6, all we have left is what? 7 and 8, lay them straight. Okay, remember tomorrow is a review, Thursday I'm just open at lunch, and Friday is a review. Okay, yeah, summer sun. Okay, make good choices, have a piece of toast.